All right, now I'm going to start on page 80 in your lab manual. This is inoculation of agar plates. We're going to start it this week. We'll look at them in a lab that comes up the next week. But the idea here is that we're going to do two different tests using these things that you might have seen before as a petri dish. And if you look really closely at the petri dish, you'll see it's got some like jello-y stuff in it. That's LB agar. It's like jello with nutrients to grow bacteria or fungus. It's like yummy stuff, right? So we're going to do a couple different tests. These two plates in particular have just plain old food for bacteria on them. And if you go to page 80, you will see that if you were in lab, you and your group of 4 would get to divvy up one of these plates by making an X on the back of it, on the plate. It would actually look like this. See how there's an X drawn on it? And then what would happen is each of the 4 people, like me, teacher, Boo Boo Bear, Danny Dog, and Susie whatever, would get to have one of these corners. They would take what's in here, which is a sterile cotton swab, and they would decide to swab some portion of the environment. It could be like the underside of this cabinet. We could go outside and sample the surface of plant leaves. We could do a light switch. All these, and then you rub your little cotton swab on that spot, and then you rub it on the plate. You let it grow for like a week, and then you come back and you check it and see whose spot had the most bacteria, the most exciting stuff. And I do have an ant crawling down my arm. So that's part 1, is we make the little cross, we pick the different environments to sample, and then we let it grow and come back later and see which grew the most. I'm just going to let you know now that I'm going to tape the sampling and doing this on the plate on a separate video, because I'm going to go outside. We'll do some exciting places that aren't so super duper boring. The other experiment we do with these dishes is on page, if you turn the page, page 82. You'll see that you are going to have 2 more plates. These 2 plates, that's these ones with this little blue line, have that LB agar, so the food for bacteria, but they've already been coated in a layer of E. coli bacteria. This isn't pathogenic. It's not the kind of E. coli that makes you sick from food poisoning. This is the kind people use in the lab all the time to test things out. It's not bad for you. What we're going to do is see. We've got 4, I'm going to point the camera down so you can see. I've got 4 different substances here. I have hydrogen peroxide. I have ammonia. I have this stuff called cavicide. And I have hand sanitizer. And what I'm going to do is I'm going to soak one of these little paper rounds. It's like a little paper hole punch. I'm going to soak it in that substance, and then I'm going to put it on that plate, and I'm going to see, the goal is to see, say hydrogen peroxide, can it stop the growth of E. coli bacteria? Can ammonia stop the growth of E. coli? Can this cavicide stop it? Or what about hand sanitizer? Can it stop the growth of the bacteria? So what you want to try and do up front is guess, make some hypotheses. Do you think any of these can stop the growth of bacteria, yes or no? And then the other thing to note is that a couple of these things are actually what are called disinfectants, and the other ones are called antiseptics. So a disinfectant is going to be a substance you use on non-living tissue to clean it. Say, for example, ammonia is a popular as a cleanser. Other of these are going to be antiseptics. That's what you use on living tissue to sterilize it. Iodine is like an antiseptic, but hydrogen peroxide also. So it's kind of like we're separating these up as well. And I will do a little video to show you how I'm going to put the little paper doodahs on the plates and label them up and do that properly. The second plate I make here is going to test 4 different types of antibiotics to see if they can stop the growth of this bacteria. And the antibiotics I have are, I believe, erythromycin, ampicillin, tetracycline, and one that starts with the letter N. What is it? I think it might be novobiocin. So one plate will be these 4 things. The other plate will be 4 different antiseptics. And I'll set those up now. Okay, first off, I'm going to do the antiseptics and disinfectants on this plate. You'll see that I've labeled the plate not on the lid, which is at the top, right? But instead on the bottom, because if I take the lid off, and I won't know which is which on where I did anything. So you always label the bottom of your plates like this. Take the lid off, the hydrogen peroxide, maybe even pour a little bit into this beaker. Teeny tiny bit, I don't need much. And then I'm gonna take my little tiny paper doodah, soak it. If you can see that, I'm just going to soak it in there really quick. Hopefully I can pull it out somehow. Don't float away. I don't want it to be too soaking wet, just because that might run all over the plate. And then I'm going to do is pick this up and just drop it and give it a tap, tap, tap. That's all you have to do. Next up is ammonia. See if I can do it this way. Ooh, it's stanky, just like ammonia. And ammonia's right here. So you'll see it every time I just drop the little dot onto the plate and flip it back over upside down. This is the cavicide. And I've got a new set of forceps every time so I don't mix up my substances with each other. And you can even see it's kind of watery, so you want to be careful. And then, last one here is hand sanitizer. This will be fun, right? This stuff's kind of gooey. Ooey and gooey. I'm just trying to get because the hand sanitizer has so much gelatin on it, I'm trying to make sure I don't put too much on. And that's it. Now I made my first plate. Put all this stuff back. Next step will be antibiotics. Okay, next up, I'm putting these antibiotic discs. This is a fancy way I've not done before, but we'll see how it goes. Onto the plate, like that. I'm just hoping it doesn't fall off. Try to tap it into place. See, it has a little E on it, so that's erythromycin. Maybe you can't see that. Next, I'm going to do tetracycline. Oh, this thing's around backwards. Hold on. Hold on, people. Fun times, right? Okay. There we go. Tetracycline. Got this Novobiocin. Oh, this one's actually set up properly. Yay! Novobiocin. Oh, put it in the wrong spot. Terrible. Let's move you over. We'll have to remember there's a little, that's where the Novobiocin fell. And then we've got, what is it? Ampicillin. That's the last one. There we go. Ampicillin. 1, 2, 3, 4. So I'm going to incubate both of these plates for a day, basically at room temperature, and then I will keep them. I'll take some pictures of them and help you guys figure out what happened.